in terms of oxidation reduction potential or redox potential. So they require a negative redox potential. And that implies that oxygen is removed from their immediate environment to enable viability, then enable their metabolic activity, and also to enable them to multiply. And what happens is that when they are exposed to oxygen, they carry on some biochemical reactions, all right, but to a limited extent, because the byproducts of those reactions are toxic to the organisms. When they do them in the presence of oxygen, there's some products they can't deal with, can't just inactivate, and those substances end up being toxic and destroying them. There are several of them. Among these toxic products, we have negatively charged superoxide radical O2 minus, and we also have hydrogen peroxide. So this would accumulate and destroy the organism that released them. So even their energy producing reactions, these are fermentative pathways, and they get energy using some other compound to act as the oxygen, the electron acceptor at the end of the exercise. So they don't use oxygen, and they utilize the fermentative pathways. You're familiar with the byproducts of fermentation. So they are different naturally in that they lack some properties which are possessed by strict aerobes and facultative anaerobes. These properties include the cytochrome system, especially the oxidase enzyme, which is totally lacking, and also other enzymes are lacking. Catalase is one of them. Peroxidase is another, and superoxide dismutase is another enzyme that is missing in these strict anaerobes, but is present in aerobic organisms and facultative anaerobes. Now, these enzymes have their roles, and like superoxide dismutase, it catalyzes the conversion of superoxide radicals that are released in the aerobic organisms and facultative anaerobes. They have that capacity to break that down because they have the enzyme superoxide dismutase, and it catalyzes the conversion of superoxide radicals to hydrogen peroxide and oxygen. And hydrogen peroxide also requires further breakdown, and catalase breaks it down to hydrogen, to water and oxygen. So these products don't accumulate in strict aerobes and facultative anaerobes, but because of the lacking enzymes in the strict anaerobes, they accumulate and the destruction of this organism. Otherwise, it's a large group of bacteria, and usually when you have a large group, they are extremes. So you get one extreme of organisms which are strictly anaerobic. One can describe them as strictly anaerobic. And at another extreme, you have some which can withstand a little oxygen. And it stops that they might have some small quantities of these enzymes that are necessary, but they are able to withstand it. They can remain viable, but at a low grade level. When oxygen is totally removed, then even if it's growing, they grow better and the colonies become more permanent. And being a continuum, you have several in between. There are some right up in the middle, there are some which are leaning towards, there's very strict ones, and there are others which are leaning towards the ones which can tolerate a little oxygen. So that whole group can be divided into very strict ones, some which are not so strict. And if you read some details, books which have details of microbiology, you might have those divisions described, but we'll just put them together and discuss them as a group, bearing in mind that they are organisms which are affected negatively by oxygen. So the organisms we are talking about, you can mention them, and to mention something better, it's better if classification is done, so that you can know which class or how to mention them. So classification and examples of anaerobic bacteria associated with human infection. So they are bacteria and they're classified like other bacteria, using applying all those methods that we mentioned that are applicable when one is classifying bacteria. But we are going to look at this using one method, the commonly used one based on Schur, Ramstein reaction, and spore formation. So if you look at the shape, they are bacilli, which are anaerobic bacteria, which are strict anaerobic. And they are bacilli, and based on Ramstein, you have gram-positive bacilli, and based on spore formation, they are non-spore formers. So they are described as bacilli, gram-positive, non-spore formers. And there are quite a number of genera in that group. One is Actinomyces, then there's the bacterium, there's bifidobacterium, and there's Propionibacterium. Secondly, other bacilli which are gram-positive but spore forming. And we are going to elaborate on this group. The main genus here is genus Clostridium. And then there are gram-negative bacilli, which are anaerobes or strict anaerobes, and the gram-negative bacilli usually are non-spore formers. They are not associated with spore formation. So you just have gram-negative, non-spore forming anaerobes. And there are again very many genera there. Examples given there are Bacteroides, Privotella, and Fusobacteria. Then there are cocci also in the group of strict anaerobes. And they are gram-positive cocci. These are like staphylococci and streptococci, but they are strict anaerobes. So one genus is called Peptococcus. This one corresponds with staphylococci, which are facultative anaerobes, but these ones are strict anaerobes. And then there's Streptococcus, which are like streptococci, but they are strict anaerobes. Then there are also gram-negative cocci, which are strict anaerobes, and one genus that is known, is referred to as Veillonella. Then spirochetes, you cover spirochetes later, and you learn that they are actually not cultured most of the time. So they are not easy to grow on media like cocci and bacilli. These spirochetes, there are some specific species and strains of a genus known as Triponema and Borrelia, another genus, and these require oxygen-free environment for propagation. And some of these are also culturable, but all in all, spirochetes, most of them are not culturable, but these specific species, when they are being kept in the laboratory, they require oxygen-free environment. And should one encounter the culturable ones in this group, they will be incubated in an environment that is free of oxygen or air. Air contains oxygen. So this is just for completion, but spirochetes in general, it's a broader group of bacteria than that, but just to mention that there are a few where this lack of oxygen is relevant. So where the strict anaerobes found, the occurrence generally they occur in the gastrointestinal tract of animals. They form part of the normal flora in these animals in their gastrointestinal tract, uh, food in the mouth, food in the large gut, and then they're also found in the environment. There are some which may be in the environment naturally, yes. Environmental bacteria, which are strict anaerobes. But there is a group that features the environment from the gastrointestinal tract of animals. They are passed out. When the animal passes out the feces, these organisms also come out into the environment and they can be found in the environment in parts like the soil, water, and also in sewage. Then if we narrow down to humans, the organisms occur in normal flora. They are present in the mouth, quite a number of them, in the large intestine, especially in the lower parts. In lower parts of the genitourinary tract, there's one portion that has a small number of normal flora, especially in females. These organisms may be found there. Some of them in females might originate from the large intestinal tract due to prostate, and they occur there as normal flora. Not prominent component of the normal flora there, but from time to time they are detected in small numbers. Also, some specific areas in the skin, especially those that are close to the openings of the areas where they are found. Yeah, my internet might start misbehaving. So they are also found in various tissues in association with disease. So that one is not natural, but from time to time, they cause disease and can be isolated from the infected tissue. So we mentioned that these organisms are handled differently when it comes to the laboratory part, and they are the laboratory techniques are restricted to these organisms for detection of strict anaerobes. Like other bacteria, they can be demonstrated through staining and microscopy. And if you remember, we have just classified them according to other characteristics, including staining and what is seen. So gram stain and microscopy for most organisms. Secondly, another method of detecting their presence in a specimen where this particular technique is available or identifying them. This method is called gas liquid chromatography, where the solution containing the organism and its products can be spread using the technique of chromatography. It could utilize a specimen for detection of products of metabolism. Among these, we have butyric acid, propionic acid, and oxybutyric acid. So when a specimen is run through this chromatography, there's a way those who are handling it can tell there's butyric acid in it, or there's propionic acid, or there's oxybutyric acid. And from there, one can conclude that there is a strict anaerobe in that specimen. And some people can go further and say which particular one. They know the patterns, how much propionic acid is produced by such an organism, and it's butyric acid. So they can stretch it further to identify the most likely strict anaerobe in that specimen. But first, it shows that there's a strict anaerobe, and secondly, it might give them an idea which one. Then there are special culture techniques for isolating these organisms in the laboratory. This could be from a specimen or just growing them in the laboratory. So these culture techniques are a variety of methods. They are either improvised or designed to create suitable growth media and atmospheric requirements for incubation. And then specific genetic material detection and analysis is also applicable. The molecular method can also be used to detect these organisms or to identify them. So if you look at the culture methods for strict anaerobes, first you need a specimen. This we are going to tackle on the point of a specimen because we are thinking of the diagnostic approach, how to do it. So there are some specimens which are more likely to contain strict anaerobes than others. So for these ones, whenever they are received in the laboratory, those people who are handling them always data for these organisms as well as the others. So there are higher chances of isolation from specimens. These could be from one, infected internal organs, other tissues, cavities and fluids. So this is infected internal tissue. It could be an organ, or it could be any other tissue, or it could be a cavity. Cavities are like pleural cavity or pericardial cavity or peritoneal cavity. So the fluids there. So for where there's an infectious process involving any of these, the material that is obtained for analysis, one always looks for strict envelopes in addition to other organisms. So the type of specimens could be attributes, fluids from various tissues, could be a discharge of pus from abscesses, or it could be samples of infected tissue itself. So those ones are processed for strict general as well as the other organisms. Also, specimens from superficial or deep pools, and also if they are contaminated with dirty material, this could be soil or they could be containing dead tissue. So after obtaining the specimen, the way to collect it is important. It's collected carefully and transported in a special manner and processed, applying necessary precautions to increase chances of isolation. These organisms sometimes or most of the time are in small numbers. So if the specimen is contaminated, the contaminant might predominate, might overshadow them, and they can go undetected. And exposure to air, we have emphasized that they are sensitive to the oxygen component of air. So they should not be exposed to air. They also dry up very quickly, so that sample should not be allowed to dry. They are delicate organisms. They dry up if the sample is exposed to air or exposed to dry conditions. So the culture media, BA is useful here. It has adequate nutrients for most anaerobes associated with human infections. And it is advised that it should be freshly prepared, and if it is to be prepared and stored, then it should be stored under reduced conditions where it is not exposed to oxygen. Then the selective medium also, if this is needed, and it is BA prepared with some antibiotics to make it selective. And there's a third medium called Robertson's cooked medium, abbreviated as RCM. This is a medium which contains minced meat, pieces of minced meat, and this is suspended, mixed with fluid, which has a lot of nutrients in it. And there's a way this will be explained to you through the practical. Now that meat part of it creates an environment without oxygen. There are some reactions that take place and oxygen is reduced or removed completely. So its place in the laboratory work for strict anaerobes. One, it can be used for initial inoculation of specimens which may contain strict anaerobes in small numbers to enhance their multiplication and isolation. So in this sense, it acts like enrichment medium. So part of the specimen is inoculated to enable the few organisms to multiply, to increase in number. And when they are more, then the chances of picking them and culturing on a semi-solid medium are increased. So initially, inoculation is done and incubated for 72 hours. Then subcultures are made on BA and incubated after 2. As another use is maintaining these organisms in laboratory, that is propagation. So the medium is inoculated with organisms which have been isolated, and one needs to keep for reference or for use. So it is put in RCM, and maybe from time to time it is transferred to a fresh. Then fourthly, there's a medium called glycolate medium, and it contains the reducing agent, sodium thioglycolate. So that's the medium, supportable. Then incubation methods of these cultures. They are improvised and designed to ensure complete elimination of oxygen in the incubation environment. The most commonly used method in many laboratories is the jar techniques, and they are referred to as anaerobic jar technique. One of them, you might have seen this during your introductory part of the protocol, Fields Macintosh or Macintosh Fields jar. As the name implies, it's a jar made of metal. It has a heavy lid and it has valves, one for inlet and another one for outlet. So for the outlet, air is sucked out, that is evacuation of air. And for the inlet, that air, a mixture of gases is pumped in. Now this is done when the culture plates are already they have been put in the jar, piled there, then one places the lid and sucks out the air in the jar through this evacuation, and then replaces that air in the mixture of inert gases. And part of that mixture is carbon dioxide and hydrogen. So these become clearer when you see them, how they are used. Then another type of jar is called gas pack or soil jars, and these are named after their manufacturers. So they are different from Fields Macintosh jar in that they are made of some plastic or material which is transparent, and it also has a lid. And instead of pumping in air and evaporating, it has uh, chemicals in a powder form packaged in sachets, and these are included. Once one is ready to close it, the sachet is cut and water is added to this chemical and some bubbling that generates the mixture of gases that is required. And that mixture still includes carbon dioxide and hydrogen. So in both jars, traces of oxygen is made to react with hydrogen, which is part of the mixture of gases, and this reaction is catalyzed by palladium. This palladium is a part of that the inner part that contains the palladium. It's a catalyst to facilitate this reaction so that all the oxygen that could be there is utilized, of course. Then there are many other improvised methods. The chambers, which are for strictly anaerobic chambers. There are other methods which utilize metals that get oxidated. There are others that use some organisms that can consume the oxygen, but the easiest and the commonest one is the jar technique, and maybe the chamber, the anaerobic chamber method. So with that introduction, we can start on some of these organisms that are strict anaerobes. And we mentioned them when we are giving a classifying and giving examples. The one genus there we mentioned as Clostridium. And it consists of gram-positive spore-forming bacilli. And most species in that genus are flagellated and motile. Most, that means there are exceptions. Some of them are not, but most of them are motile because they have flagellum. So as a spore-forming organism, it has spores, and this spore, when once is a bacillus that has a spore in it, the diameter of the spore is larger than the width of the cell. So you have a bacillus, if you remember, these are rectangular shaped organisms with the length and the width. If you have part of it with something bigger, it will cause a distension in that part where it is located. So that is what we mean by the diameter of the spore is larger than the width of the cell. So this is basically to differentiate it from another gram-positive bacillus that forms spores, that is the genus Bacillus. But these ones are, the diameter is larger, but when it fits into the bacillus, here it causes distension where it is located. And its position on that bacillus, it could be at the center, it could be close between the center and the end, which is subterminal, or it could be at the very end. And that is described as terminally placed spore. Now these spores, they are typical bacterial spores which are formed by these organisms to enable survival during hard conditions. So they are resistant to adverse physical conditions and they enable prolonged survival of the organism and more so in the environment. And so these organisms are found in the large intestinal tract and they are passed out with the fecal matter into the environment, and then they form spores there and they can survive because of that spore for a long time or a longer time than other bacteria. So it enables survival. Now that is the genus in general. And when you come to the species, they occur as normal flora, again, in the intestinal tract. And they're also in the environment, like what we have said from the strict anaerobes. It could be found in the soil, in water, in decomposing animals and plants. They contribute quite a bit in the composition of these animals. Now, the species of Clostridium are many. There are some, and many of them can be associated with human disease. Among those which are associated with human disease, one is called Clostridium perfringens. This is also known as Clostridium erythrae. It's the same organism. Then there's Clostridium tetani, there's Clostridium botulinum, there's Clostridium difficile, there's Clostridium histolyticum, and these are a few examples. There are more species and more species which are associated with human disease. Then there's one species, Clostridium sporogenes, It may be isolated from specimens. It could be isolated from the environment, but it's not involved in disease position. So for the pathogenic ones, pathogenic species of Clostridium, they have some properties which enhance their pathogenicity, which increase their pathogenicity. Yes, allow me to connect. So they have some properties which one can put a finger on, which strengthen their capacity to cause disease or increases their pathogenicity. For most pathogenic species, they share a few. One of them is the presence of spores. The spores, we say, enable survival in the environment or in harsh conditions for a longer period. And when something survives longer, then in this case, the spores, the chances of a susceptible host getting into contact with it are higher. So the spores increase the chances of survival, and as a result, they enhance transmission because by increasing the chances of that possibility of a susceptible host getting into contact with it. Then if it died faster, then that chance is lower. But if it's given a longer time, then during that interval, an animal or a human being can get into contact with it. Then secondly, by the fact that they cannot withstand oxygen, that is the ability to retain viability and multiply in tissues without oxygen or reduce blood supply. This is important because there are some illnesses or infections which are related to these characteristics by virtue of them being anaerobic or unable to remain viable in tissues without blood supply or without oxygen. That increases the other organisms would also become non-viable if they depended on oxygen. But for these organisms, they remain viable and the chances of causing disease is increased or their ability is strengthened. Then the third one, these organisms produce exotoxins. So the pathogenic Clostridium species, their main virulence factors are exotoxins, which they produce. These are responsible for major clinical manifestations of associated diseases. Now, these exotoxins are different types, their composition and mechanisms of action differ among the species. So those three characteristics are common to the Clostridium species, which are associated with disease possession, in other words, pathogenic Clostridium species, in relation to disease possession. But in addition to that, this one is not general. There are specific pathogenic species which produce addresses and other enzymes, which we talked about when we did pathogenicity virulence. So that is now specific, it's not general. It's just some and not others. So if we go to one of the species, Clostridium perfringens, also known as Clostridium wildtribe, it contaminates the environment, as we have mentioned, because it originates from the humans and other animals. And for this particular species, it's recognized by the spores being present in the middle of the bacillus. And it has the capacity to form the spores also while still in the large intestinal tract. So if one analyzes the content, one might see Clostridium perfringens spores in the content, and these spores can also be passed out together with the vegetative form in the fecal matter and into the environment. So it forms spores in the intestines and it forms spores in the environment. And it's an organism that has a capsule, it's capsulated in tissues, and it is non-motile. So this is an exception. We said many of them are flagellated and motile. Clostridium perfringens is non-motile. Then it has some significant biochemical properties. One of them is described as saccharolytic activity. It has the capacity to break down carbohydrates. That is the predominating biochemical property, and it has little ability to break down proteins. So it is mildly proteolytic. And it can also reduce nitrates. Nitrate reduction test is positive for this organism. Then it has antigens, which it releases, and they are different antigens designated with capital alphabetical letters A up to E. Because of these antigens, it can be classified further into serotypes. The one that has antigen A is serotype A, one which has antigen B is serotype B, one which has antigen B is serotype B, etc. Then virulence products of Clostridium perfringens, we have mentioned exotoxins. We are talking about pathogenicity, virulence and pathogenicity go hand in hand. So exotoxins are major virulence products. Several different types listed by the serotypes of Clostridium perfringens. And these exotoxins are responsible for tissue damage and severity of disease manifestations. And each serotype releases more than one exotoxin. And each type of exotoxin can be released by different serotypes. In other words, they are overlapping hybrid. But each exotoxin has its specific mechanism of activity, which is different from the other. So they are unique in the way they act, depending on which exotoxin one is talking about. So there are many exotoxins. Among them, there is one called alpha toxin. And it is released by all serotypes of Clostridium perfringens. And it is a major toxin released by Clostridium perfringens serotype A. It is a toxin which is relatively heat stable, but it is, although it is an enzyme phospholipase or the CPase, but it's relatively heat stable. It's not damaged by heat as fast as other enzymes. And its main action is that it splits lecithin, which is the constituent of cell membrane. Another toxin released by Clostridium perfringens, which is significant, is an enterotoxin, but it is not released by all members of the species. There are some specific strains of Clostridium perfringens that can release the enterotoxin. So apart from exotoxins, there are other virulence products, and this is one example of a pathogenic species with those other virulence products which are unique to it. We have mentioned it's being encapsulated, and it also releases various enzymes like Anaerobic. So the clinical implications of Clostridium perfringens, it is associated with several clinical conditions. One, it can infect the wound, and it can also cause other soft tissue infections. When one looks at wound infection and soft tissue infections, there are two types. One, it causes a condition called gas gangrene. This is related to wound and other soft tissue. And secondly, it is like other pathogenic bacteria and pathogenic bacteria. It can cause wound infection and other soft tissue infections in general, like those other organisms, which is not gas gangrene. Secondly, it causes food-related gastroenteritis or food poisoning. We just talked about an enterotoxin being produced by specific strains, and these are the ones that are responsible for this foodborne illness, gastroenteritis or food poisoning. Thirdly, it's associated with infections involving pelvic tissues in adult females. In other words, infections which are related to reproduction, female reproductive system. And then it is also from time to time associated with intra-abdominal infections. That is, infection which occurs outside the intestinal lumen in the peritoneal cavity. Then we've mentioned this condition called gas gangrene, and it is also referred to as clostridial myonecrosis. And if you look at that myonecrosis part, it's something related to skeletal muscle. So it is a condition associated with rapidly spreading swelling, necrosis of tissue at the site, and myositis, that is inflammatory process involving skeletal muscle, and also gangrene and evidence of gas in involved tissues. So there are some predisposing factors to this condition called gas gangrene. One is traumatic injury. In other words, with a wound that is treated, most of the time it is being treated by trauma. So traumatic injury, it's severe enough to cause destruction or injury to the muscle, skeletal muscle that is in that site. Then the injured tissue, the soft tissue, might also be exposed to, most of the time, if it is exposed to dirty material, the chances of developing gas gangrene are higher again. So you have traumatic injury with skeletal muscle damage, then that injured part, including the muscle, is exposed to dirty material. We're going to mention the kind of material we are referring to, and then injury can also occur as a result of injury involving the abdomen, especially where that injury causes damage to the large intestinal wall. So if that is damaged, then there's leakage of intestinal contents, which contain the organisms from the lumen to adjacent tissues. So one will end up with this condition developing in the abdomen. So the organisms we are talking about, which are regarded or which are encountered as positive agents, we are discussing Clostridium perfringens is one of them, and it's the one which is commonly associated with it. But mostly it is a condition which can be caused by Clostridium species, many different ones, as a single organism or more than one organism at the same time. Because we have talked about exposure, it just depends on the species that would be exposed. If there are multiple, then you get all of them involved. If it's one, it will be one organism. And if there are other organisms, they might also have a role to play. But more commonly, we have Clostridium perfringens. The most commonly isolated positive agent and more frequently seed type A. Then the other species, there's many other species of Clostridium which are capable of causing gas gangrene. Then there are facilitating factors in its development. We have touched on this, but we can put them into some perspective. Prevention injury has been a facilitating factor because it creates adoption and it causes damage to skeletal muscle. And when these are exposed to contamination with dirty material, this dirty material, it should contain the spores of the bacterium or the species that would end up causing gas gangrene. Another important facilitating factor is the impairment of normal blood flow. Again, this is due to injury. When the wound is being created, the skeletal muscle is being damaged, also blood supply to that area is being damaged. So impairment of normal blood flow, and that leads to reduction in oxygen tension in the blood. And then thirdly, devitalization of tissue. So due to lack of blood flow and oxygen, the tissues get revitalized. And when it gets no oxygen, no blood flowing in tissue, it means there's no oxygen. And then in addition to that, you have revitalized tissue, and together that would create a suitable condition for viability of Clostridium. So that is how injury is a facilitating factor. Then the spores, they should be present in the dirty material or the contaminating material. And they get into the wound and they germinate into bacilli, which multiply. And as they multiply, they release the exotoxins. So the effects of those toxins, tissue destruction and gas production. And among all the exotoxins, the alpha toxin is the major cause of the tissue damage and severity of the medical disease. It's the major, that means there are some which are minor in terms of tissue damage and clinical manifestations. That is the key factor in the destruction of the manifestations of the gas production. It's all due to the alpha toxin. Then the foodborne gastroenteritis due to Clostridium perfringens. This is associated with food contaminated by specific strains. These particular strains, they might not be so common, but they have been identified. They form spores which are different from the other spores. These particular spores survive boiling for several hours. So if food is contaminated and it's boiled in the way it's usually done, then it still remain viable. And the food germinated to bacilli in the food, and then the food is eaten with the bacilli. Somehow they pass through the stomach and the small intestine, and the food is consumed. And then when they reach the large intestine, they release an enterotoxin. This happens when they are converting themselves into spores. You see the spores are formed in the large intestine as well. So they release that enterotoxin and one ends up with the manifestation. So the manifestations occur 8 to 12 hours after ingestion. The majority are mild. illnesses. So they are characterized by mild abdominal cramps and diarrhea. Fever and vomiting are uncommon, and like other food poisoning, it subsides on its own within 24 to 48 hours. So maybe because of diarrhea, one might require fluids to be taken, but it's not a severe illness as such. And should there be a need for laboratory workup, laboratory investigation, one needs to take diarrheal stool specimen and also the leftover food and isolate this organism. And it is very important that one has the capacity to identify these specific strains because stool in general would definitely have some Clostridium contingent. So one has to go a step further and identify the specific strain which form the spores which are not easily damaged. So that is the challenge, and now it comes to laboratory diagnosis. The capacity to identify the specific strains of Clostridium perfringens. So there are also other infections which we mentioned when we were making a list. Those associated with female reproductive system, they can occur as complications associated with reproduction depending on where that activity of childbirth takes place, the cleanliness of the environment, whether it is traumatic or usually delivery can be traumatic and maybe it was not expected, it can occur anywhere on the road, chambers, everywhere. So the mother could get exposed to that organism from the environment. That delivery is taking place. But it is also associated with traumatic abortion, especially when these are carried out under unhygienic conditions. So the organism originating from the large intestinal tract can be transmitted to the vitalized tissues also. So it could come from outside, it could come from the person himself through contamination directly or by used items if they're contaminated. Intra-abdominal infection that we have mentioned about the large intestinal tract, it's all brain damage and coming out of it from the lumen or containing whatever contains spilling over other tissue. And then wound and other soft tissue infections, we have talked about one type, that's gangrene. Then this other type is the general type, like wound infection and other soft tissue infections. Not a common cause of wound infection, but it's capable of causing wound infection, just like any other organism. But it is not, it is an infection that does not involve the skeletal muscle, it doesn't involve gas formation, so it is not gas gangrene. So for laboratory investigation and gas gangrene and other infections like Clostridium perfringens, other than the food poisoning, that one is not really an infection, but the other soft tissue infections, female reproductive system infections. The specimens are many, depending on the manifestations. It can be in a swab form, weight form, tissue form, but we have mentioned the significant, the way that specimen should be handled. So collection, avoiding contamination, and the necessary exposure to air, and then transported in a similar manner, also from wind. So it is the way it is handled. Then the procedures, gram stain or specimen, and culture for isolation and identification. So gram stain, if it is straight from the wound or from the fluid or tissue, one is just say gram positive bacilli. Then culture is performed. We have mentioned the media that I use, the way they are incubated, and then colonies, they are identified. So the media, the main one is EA, and then that is part of the specimen is seeded directly onto DA, but a portion of it is put in RCM initially before they put in DA. So incubation methods we have described again, and temperature 35 to 37 degrees centigrade for about 48 hours. They will take a little longer to grow than other bacteria. Then the colonies of Clostridium perfringens, these are different from the colonies of other bacteria that they have a double zone of hemolysis. So in terms of size, we described as being relatively large. They are translucent and they have a smooth, shiny surface. Around each colony, there are two zones of hemolysis, and that is termed double zone hemolysis. There's an inner complete, an inner zone of complete hemolysis, and a larger outer zone of incomplete hemolysis. And these are produced by the various toxins or enzymes that the organism produces. Then one stains the part of the colony, and in young cultures, one is expected gram-positive bacilli. Then with further incubation for a little longer, one later be able to see the position of the spore of that bacillus. Then if one is dealing with gas gangrene, it's important to go a step further and demonstrate the ability of that isolated organism to produce alpha toxin. And it is a presumptive identification test, and there are many methods of doing it. One of them is called NAGLA test or NAGLA. And how it is performed, you can read about it, you can have it demonstrated to you in the practice. Then there are different ways of managing gas gangrene by different people, but among those methods, one is specific management. Here, surgical approach is important, especially in removing dead bacteria, dead tissue from the wound. Should be removed as much as possible. It's what contains the organism, it's what facilitates multiplication and worsens the disease. So surgery is very significant here. And it can even be done repeatedly. And in addition, there is administration of effective antibiotics. Commonly used are penicillin and metronidazole. Some people use single penicillin singly, but some people combine penicillin and metronidazole, and there are also other antibiotics which we learn about in the clinical years. There are supportive measures that vary according to the patient. Then the aspects of prevention, cleaning and removal of foreign materials and dead tissue from wounds as soon as they are encountered in a healthcare institution. In general and specifically traumatic wounds. Then disinfection of skin before invasive procedures and use of sterilized instruments and materials, especially when dealing with invasive procedures. Then antibiotic prophylaxis, this is given as part of management when one encounters a fresh wound. After it's clean, whatever needs to be done is done, but that person is usually given some antibiotics. yes, and single injection. It depends on what is used in that particular scene. So management of fresh wounds or trauma associated wounds is part of it. Is administration of like such prophylaxis. Then the other species, Clostridium tetani, this particular one is flagellated. The bacilli are flagellated and they are motile. And they are found in the large intestinal tract like the others, in humans as well as in other animals, including horses and donkeys. So the feces of these animals can be a source of infection in some specific areas where they, like donkeys are used. Not all the feces have that infectious form. It's just good to know that some of these fecal matter, even cows, cattle, feces from cattle, could be a source of infection to humans if exposed. So that is how it finds its way into the environment via the feces. Its significant biochemical property is that it breaks down proteins. So it is predominantly proteolytic. And morphologically, its core is observed as a distended part at one end. In other words, it's an animal's core. If one is looking at one bacillus, you see a distinction at one end, and that has earned it a lot of descriptions. Some people compare this to a teaspoon, others call it, see it as resembling a tennis racket. And most books have it described as drumstick appearance. There are some sticks, there are many types of sticks for beating drums, but there are some that are shaped like that. So they are called drumstick appearance. So significant physical properties of Clostridium tetani spores. Again, as a species, there are many strains. So the physical properties would vary according to the particular strain of Clostridium tetani. So one, there are some strains which form spores which are really inactivated by commonly used physical methods of treatment of items. That is sterilization methods and disinfection methods. But on the other hand, there are some strains which form spores which are highly resistant to physical conditions. In fact, some of them are the most resistant spores ever encountered. So these resist commonly used methods of disinfection, resist adverse environmental conditions and how they change, and therefore they are capable of surviving for years in the environment. They also resist boiling. One can boil, and if that boiling takes place for less than 3 hours, then some of them will still be viable. So here, it's hard to boil something for 3 hours because most of the time it boils and evaporates. I don't know whether it's one keeps on adding, but it's just to illustrate the resistance of these particular spores. Then fourthly, they can resist dry heat at 150 degrees centigrade for 1 hour. So sterilization by dry heat is usually done at 160 degrees because of this reason. Is somebody putting on music or what? So the extracellular products of Clostridium tetani, mainly one is called tetanolysin. It is actually a hemolysin of Clostridium tetani. So it is called tetanolysis. It causes lysis of red blood cells, and its effects are mainly observed in cultures. Uh, it's not significant when it comes to clinical manifestations of the disease. It's detectable mainly when the organism is grown in a culture medium containing intact red blood cells that undergo lysis because of this tetanolysis. Secondly, and more importantly, is a product called tetanus plasmin. And this is the exotoxin of Clostridium tetani, and it is the virulence factor essential for disease manifestations in humans. And it is a neurotoxin. And it is also that component that can be extracted and be converted into a toxin. So we talk of tetanus toxin. Initially, it was the tetanus plasmin treated in a special way that would enable it to lose its toxicity, that retain its antigenicity, or its part that stimulates the production of antibodies. And that is the tetanus toxoid that is useful in immunization. So clinical, there's something covering my screen, but I believe it's clinical implications of Clostridium tetani. The illness associated with it has been there for many years, and it's known to many people. It is called tetanus. Most infections by Clostridium tetani are acquired through the spores. Its spores getting into an organism. These spores may be derived from contaminated foreign material. They could also be directly from parts of the environment. These foreign materials could acquired also from the environment. So it could be found in soil, dirty clothes, dust, instruments, and other items. So it gets into the wound, and in the wound, the spores germinate into bacilli, and this will be facilitated where there's reduced oxygen tension and presence of devitalized tissue. And once they become bacilli, they multiply. And exotoxins are produced by live multiplying organisms, so they release the tetanus plasmin. The organism remains confined to the wound, does not invade further to the surrounding tissue or deeper, or it doesn't get into the blood circulatory system. So as they multiply, tetanus plasmin is released, and this is the toxin. It diffuses into the blood circulatory system and eventually passes through nerve trunks into the central nervous system. And here is where it exerts its activity. It prevents the affected neurons from releasing the inhibitory mediators in motor neuron synapses. These inhibitory mediators are usually well known by students. Now you know their names, but they kind of regulate neurotransmission. They control it in a way, and if they are not released, then there's no control with motor neuron synapses. So as a result of that, there is overactivity of the muscles in response to stimulation. So even a little stimulus would make them overreact. So overall effect, generalized, strong, sustained contractions of muscles or spasms. And these are so strong, they are also associated with pain. They are painful. And there's also increased reflex responses to stimuli. Again, no matter how trivial the stimulating factor is, the response is exaggerated. So this infection by Clostridium tetani or exposure to the spore. It can also happen to a newly born baby. Infection of the umbilical wound of a newborn baby, and they will develop tetanus that is referred to as neonatal tetanus. So for laboratory investigations, this is easier when the wound is still present. Some people develop the illness long after the wound has healed. The toxic released by Clostridium tetani is very strong, no matter how light the infection is, no matter how little the, the toxic is, it will eventually reach the nervous system. So some of it might reach much later and the wound is no longer there, but when it is there, one can take specimens from. Especially where there's a wound that has been exposed to dirty material due to trauma, one could still get the organisms in that. So specimens and examination procedures are as described for Clostridium. So all those stages we described apply here as well. In fact, if one has that dirty wound or infected material, one might does not know exactly what is in it, so one could still find Clostridium tetani. So its colonies, when it is grown, the media the same, incubation, environment, and duration and temperature are the same. So the colonies eventually form, or the growth eventually occurs on VA, but it is not seen like those of Clostridium perfringens. There are no distinct colonies formed. And this is due to its ability to move around. It's flagellated and motile and moves all over the surface of the blood organ. So it is described, the growth is a spreading one. One call it swarming. It spreads and grows as a layer on the surface of the plate. But one can still take part of it and do the gram staining procedure. And in the early, in the young cultures, one would see straight away gram-positive bacilli. But with further incubation, then the spores would form and one will be able to see what is described as the drumstick appearance. The spores will develop and one will see the drumstick appearance. So some aspects of management of tetanus again. For this one, first and foremost, one is immune serum or anti-tetanus, that is an antibody to the tetanus toxin. It's given to that patient to neutralize the toxin. In addition to that, antimicrobial agents. And this would target Clostridium tetani if it is still there, and penicillin and its derivatives are effective. So there might be many other aspects of management and some other developments that might require antibiotics, but if we target the Clostridium tetani alone, we want to use penicillin and any other derivatives of penicillin. Then there are also other methods in the management. General supportive care, this is something that affects the muscles. The person might not be able to eat, maybe they will have a lot to jaw, might not be able to swallow. So muscle relaxants are sometimes given, and surgical management where the wound is still present. So for our part at this level, just the tetanus, antitoxin, and the antibiotics, management of the wound which we had talked about earlier in regard to student opinions. But when it comes to the actual practice, then there are other things which will be clearer when we do them. Then the aspects of prevention, again, methods similar to those applied for prevention of breast gangrene and Clostridium perfringens infection. This is because when a wound is exposed, again, I think I have said this, it's exposed to many things. Nobody knows what is in the contaminating material. So prevention is general. So the way you prevent the development of breast gangrene or Clostridium perfringens infection is the way one will prevent Clostridium tetani. So those procedures we have discussed apply. But in addition for Clostridium tetani, one is immunization is also important. And immunization is done using tetanus toxoid. And I've mentioned how this is produced. So it is given in practice as part of management of traumatic wounds. It is given during antenatal care for expectant mothers because nobody knows the environment in which the delivery will take place, and one needs to protect the mother and also to protect the one that is going to be born. In patients undergoing recovery from tetanus, this is given as they are treated. They might still go back to the same environment where they got exposed. And so they need some prevention, some immunity with them. And then it is given as part of routine immunization of children. And thirdly, use of sterile equipment and aseptic methods. So it is the one we mentioned for Clostridium perfringens still applies, but in addition, it is done in wound management, whether it is umbilical wound or fresh wound or traumatic wound. It is important that it is done aseptically, and then child delivery and care of the umbilical wound. So again, that emphasizes the environment of delivery and the way the cord, umbilical cord is cut using sterile instruments, and then the management after it has been cut. So I think we can take a break there and go to this next one at 2 o'clock. Or do you still want us to go on at least cover this one? Can you hear me?
Yes.
We can take a break?
We can.
Or we go on? We will definitely come back at 2 if we won't finish. What's your, are you tired or go on?
Break.
Pardon? What did you say?
Let's take a break.
A break. And we start at 2 o'clock. Okay?
Yes.
So. Stop sharing.